Okay, so welcome to the session, everyone. I think we don't have explicit microphones. That should be fine here in the small room. And since we are a rather small group, from the beginning on, I would really invite you to step in if you have questions or if you want to go into particular things in a bit more detail. Just go ahead. I think we are sufficiently small group to do that. And if you have no specific points, of course, we will take you basically through the basic things and tell you a story. Let's
use the speakers. Yeah, this one. Just reduce it. No. No.
Good. Okay. So Sebastian, Bludau, and myself, we are both from Jülich, and have been working on this Zebra tool suite for quite a while, and on the siibra-python atlases, and we will take you through the idea behind the atlases and these tools and try out a few things together. So we will be talking about an atlas of the human brain, which is available through the infrastructure eBrains. But this infrastructure includes also atlases from the rat brain, from a macaque brain, and also supports the L mouse brain. But today we will focus on the human atlas. Quite a few things that we will explain also apply to some of the other species here. And to go ahead, we first want to summarize to you how cytoarchitectonic mapping works, because cytoarchitectonic maps is kind of a basis of many things that we are developing here. And Sebastian will take over at this stage because he's really an expert on cytoarchitectonic mapping, much more than I am.
Okay, as Timo said, so it's mainly here for the human maps we present in our atlas. So all the maps we show, or we'll show later on in the atlas, are based on different cytoarchitectonic features of different areas. So as a starting point, we have those whole brain sections. We have a collection of 26 brains which were completely sectioned and stained for cell bodies. And those sections are fully digitized at 1 micrometer resolution, and they build the ground truth for all the maps we will see for the human atlas later on. They have a section thickness of about 20 micrometer, which is half of the thickness of a human hair, which results in about 7,500 sections per brain. For the atlas brains, we do not have every individual sections. There we have every 15th section. For the big brain, which we will also present later on, we really have every section, and all sections were stained and reconstructed. And yeah, we use those sections to look for microstructural differences of areas. So here is a typical starting example. This is the one border between the primary and the secondary visual cortex, which is, from the microstructural point of view, very easy. So on the right side, you see a very thick layer 4, which is located over here, with a lot of small cells. And on the left side is the secondary visual cortex, which looks quite different. And it's not always that easy. So in the visual cortex, in the primary visual cortex, you can even see the border with your naked eye if you look at the histological section. But if you go to higher associative areas, those borders are much finer, and therefore you need algorithms, or at least ways to quantify borders between adjacent areas. And therefore, our institute, we have developed quite a while ago some methods to really quantify those borders based on high resolution images. So here we see a border between area 11 and area SP1, which is located at the frontal pole of the human brain. And the border here is located at the green position here in the middle of the image. And this is not that obvious like the border we have seen before. And in order to quantify those borders, we draw profiles along the cortical ribbon. And those profiles can be plotted as a function here. and you can describe the shape of the function. And if you have a lot of those profiles, so we do not only draw one profile, we draw profiles along the complete cortical ribbon, and then there are a lot of statistics done to look for differences in the shape of the profiles from the adjacent areas. And if you move along this cortical ribbon and compare groups of profiles, you can calculate distances between adjacent groups of profiles, and those distances can be quantified and used for statistical tests. And if we have significant peaks at different block sizes, which mean different amount of profiles which are grouped together, then this is a very strong hint for an observer-independent border. Of course, you do have to check it in detail. Sometimes there's a blood vessel, something like this. So this is something which have to be checked manually, but at least you can verify your borders. And this border quantification have to be done in several sections. So one of the strongest hints we have that a border is real is that we can find it in neighboring sections. So because every time you will have effects on individual sections, but if there are something changing, it's the same precision, it's in a series of sections. This is a really strong hint that there's a border between adjacent areas. And since there's a lot of variability in the human brain, we do this not only in one brain, we do it in 10 brains. So all maps we will see later on are based on a set of 10 individual brains. In those brains, a series of sections were analyzed and the borders were detected in all brains. And once this was done in the individual brain, all 10 brains, we transfer them to a reference space, which is also a very important step. So all the data sets we will show later on are transferred or were transferred to a reference space, because each individual brain, of course, is different. And in order to make it comparable, we have to agree on a reference space. For the Jülich brain maps, we have the Colin 27 reference space and MNI 152, which are quite common. And once we have brought all individual brains and the delineations of the areas into the reference space, we can simply overlay them, which give us the probabilistic information. So what we here see is a probabilistic map, front to pole area. And red means that there's a very high possibility of being a specific area. So here in 10 out of 10 brains, this area was found at this position. And the periphery of the area shows the interindividual variability of the different brains. And this was done for all brains. So the maps which are included here in the Jülich Brain Atlas are not only labels, which were created somewhere, somehow. So it's really for every map and every area, there was a scientific study where the mapping was done in 10 individual brains, and there was a publication behind this individual map to make it reproducible. Yeah, okay. So here's another map from neighboring area. And this is the first step to the zebra concept, which is the main topic of the workshop today. So as Timo already mentioned, it's not only a human atlas which is included in the zebra environment. So there's also a rat atlas, there's macaque atlas, and there's just this slide here is really to show the wording we are using in the following slide. So of course we start with different atlases, which is if you have a map from different planets, for example, and then you apply a different parcellation scheme, which could be the Jülich brain, and we will focus on this one here. But it can also be something like the Lisicancliani or something else, which is comparable to countries, continents. So different parcellation schemes from different map sources. Then we have those reference spaces. So in Zebra, you always have to define which atlas you would like to use, which parcellation you would like to use, and you also have to define the reference space just to make the data sets comparable. So if you bring in your own data, you have to make sure that that the reference spaces matches. And once it's inside the Zebra environment, normally Zebra will take care of the different reference spaces and keep track of it that you're on the correct. And at last point, we have different regional features. So the The atlas consists of different maps, and for all of those maps, we have, depending on the area which is available, different features. Those can be cell numbers, receptor densities, functional, structural connectivity. This is really highly individual, depends on the area. This is also where different people can bring in different data sets and enrich the complete atlas to make it to a tool which is really helpful for the broad community, which is the general idea behind the atlas. I think you can...
Yeah, I can take over here. One comment that I would like to make, just to connect to the rest of this workshop, is what Sebastian has described is, let's say, the gold standard traditional way of mapping cortical cytoarchitectonic regions with these profiles that are computed throughout the cortex. Of course, one may ask, is it, and this is, of course, hypothesis driven, because you are already directing the algorithm to a certain region of influence where you expect a border region. And it is also controlled by an expert, right? This also makes the quality of the cytoarchitectonic atlas that we are presenting here. On the other hand, one might ask, is there a way to do this in a fully data-driven fashion? And of course, since a couple of years, we are working on deep learning algorithm that would reproduce cytoarchitectonic regions only by looking at the images, without explicit design of such an algorithm. And this is something that tomorrow Christian Schiffer, who's here in the workshop, the postdoc in my group, will talk about. Yeah, so we are doing such work in parallel. We try to do self-supervised learning and see whether the deep learning network can learn representations that cluster naturally into the same structures without explicitly knowing about the passivation scheme. And it's quite interesting that a lot of that is really coming out in a fully data-driven way, but it requires a lot of data, and it's quite a difficult thing to do. So watch out for the session tomorrow and the talk by Christian Schiffer about that. And there's another comment I would like to make on what we discussed now, that from the slide that Sebastian has just shown here, it actually results that for a given passivation, you have multiple maps, right? Because if we provide you a map of the cytoarchitectonic atlas in the MNI space or in the BigBrain or on the FS average surface, these are not the same maps, right? These are different maps, but they refer to the same passivation scheme. And this is an important distinction that we will be making here. So the passivation scheme is a concept. It describes a principle by which the passivation is computed. But in the different spaces, we of course have different maps, which are different realizations of that passivation scheme. This is something that's quite important for the framework that we are building here. Okay, so what we have been doing the last years is we have been building software services into the eBrains infrastructure, which is built here on the European side, and we have been integrating many data sets, including these passivation maps, into the eBrains as curated data sets. So the eBrains platform has different, what we call service categories for brain atlases, for brain simulation, and also for data sharing, which we are using here. Yeah. So the category of brain atlases, if you go there, you find the access to the atlases, you find tools for looking at them, working with them. You find tools for integrating data with the atlases. You find tools for analyzing atlases. Sometimes you find not a tool which probably exists because it's just a lot out there. So it's also important, if you're interested, to be in touch with us. But many of the important things should be right there, and you can find it. I would like to explain a bit now the concept of a multi-level atlas system which connects multiple scales and modalities around cytoarchitecture as a main structural basis. And the idea is here that we have passivation maps that delineate such regions and the whole brain. Ideally, this could be, and in this case, it's the cytoarchitectonic atlas. And then from there, you could get access to measures of variability of different brain regions. That's what Sebastian already showed, that we have these probability maps showing how delineations in different subjects differ. But from there, the idea is that you can also go into the high resolution original data that has actually resulted in putting up these maps. So we want to build a system where you can actually from here zoom into the histological data, get access to the full resolution data, and there even be able to zoom into the resolution of individual cells, while still having the spatial context and the spatial dimensions, which, as you saw in the talks this morning about genetics, is not always the case, right? In state-of-the-art atlas, sometimes we lose really the coordinates, the spatial dimension at some stage, in order to be able to have very deep measures. As for the genetics, here we concentrate a lot on keeping the spatial dimensions while still increasing the level of detail. Then we are integrating different modalities. So basically the chain across spatial scales that is illustrated here is the level of cytoarchitecture of cells. But of course there are nerve fibers, neurotransmitter receptors, there's connectivity and function, which we also want to link into this framework. And so the idea is also to connect the parcellations with measures of connectivity, to connect brain regions with feature distributions from histology, not only with the data that has been directly used to measure the brain regions. And if you start doing such a concept, it first results in a whole lot of data sets which have very different heterogeneous structure, different data formats, and so on. So you need software to be able to use it in a simple fashion altogether. So we have been building web viewers that allow you to actually explore the brain and to access such measures of brain organization. We have built a Python interface that allows you to program with it. And we will look at these two things. We have also built analysis tools that make use of it for certain use cases and directly embed the information in analytics workflows. That's not a topic today. And we have built a couple of tools to attach new data to the system. This is also not the topic of today's tutorial, but if you're interested, feel free to just talk with me in the breaks. So these tools here on the right, we have integrated them into a single tool suite, which we call Zebra, which stands for software interfaces for interacting with brain atlases. And the Zebra Explorer is an online viewer, which does not require any installation or software developer skills. You can just use it in a web browser. And we will have a look at that first. siibra-python is a Python library that gives you access to many more functionalities and allows you to do to code and to build reproducible workflows and scripts. And it has the same functionalities plus more. Actually, under the hood, it powers the Zebra Explorer. And there's also an HTTP API that we call Zebra API, which we have actually built to connect the Explorer and the Python interface. But it can be used for application development because it's openly accessible. An important aspect is that if you currently install siibra-python, you will find it in a version 1 something, and it is still under very active development. So there are still you will find some rough edges. Many things are working well. Some things are not fully documented. Overall, I think there's a lot to find, but that should be kept in mind. Yeah? It's still under active development. It will also warn you about that. And we are actually working on a version 2 right now, which will be much more stable, but this still has some months to go. The data that we show comes from different institutions. So the data is, besides some of them, is not coming from us, but from different institutions. And there is definitely much more data coming in. So we still have a project going, which is called eBrains 2. And there are a couple of labs in the project who get funded for integrating more data. So we're happy to discuss, and we're also happy to discuss interfacing with other tools that you might have out there in the community. All right, so we will start having a look at the online viewer. You can actually find it yourself under this link here. Atlases.ebrains.eu/viewer. That's already sufficient. if you attach go/human, you will right away be taken to the human atlas, but actually the first part is already sufficient if you want to do that. The Python stuff we will be looking at is also collected on GitHub as tutorials. Sometimes we ask participants to run the tutorials live. I wouldn't suggest to do so today, mostly because the links you find there are currently not working very well because there were several migrations taking place last week under the hood, and I saw a few problems actually, virtual machines having less memory, and so I wouldn't want to send you into that today. Maybe you can just follow it on my screen later on. But you're still welcome to try and ping me if you run into problems. All right, let's look at the viewer first. And one thing that we would like to show in the viewer is that part of the story here, how data features are linked to brain regions in a pacillation. And before we look in the viewer, I try to give you a bit more of the conceptual background. This gives more detail to the overview slide that Sebastian has put up. So in Zebra under the hood, we have a model for locations like points, bounding boxes, or even image structures are also kind of locations. So if you have a peak in an image, or let's say an activation map from a functional study, this also encodes a location. And locations are always linked to reference space. Yeah? In the human brain, we are often dealing with different reference spaces, like the free surface spaces, the M&I volumetric spaces. We talk here, of course, a lot about the big brain, which is a single subject histological reference space. And we will be jumping between these spaces when we use Zebra. So it is important that locations always have the reference space attached in order to make them interoperable and comparable. Yeah? So locations are always linked to a reference space. At the same time, brain regions, at first, they are just names, conceptual names of a pacillation scheme, and that's a semantic thing, right? This is, so the name of a brain region is linked to reference pacillation, although it still encodes a location in the brain. You know, it's just not realized in a particular reference space. It is described as a concept. Between these, there is a connection, and this connection is the brain region maps. So if we are mapping a particular brain region, let's say a cytoarchitectonic region in a certain reference space, we obtain a pacillation map in that space, and that gives us actually the connection between locations which are based on coordinates and the brain regions which are anatomical concepts. In Zebra, we can also translate between the two, so we can take a point and assign a brain region to the point, which will in the background, of course, use brain region maps, and vice versa is, of course, also possible. All these structures, which in different forms encode locations in the brain, can then be used to look for features which describe brain organization from different perspectives. And for locations in the brain, these are spatial queries. They are typically defined at the level of coordinates, bounding boxes, things like this, or segmentation masks. And we also have semantic queries, which really use the names of brain regions to search for data. Such queries result in data features, which typically has the form of a published data set, which includes files in some form, metadata, and the location associated with it that was used in the query. And from these data features, we can then typically actually retrieve data, fetch data from an online resource, which gives us concrete elements like images, tabular data, you name it, like connectivity matrices, whatever. So there's always this pattern. You have a location in the brain. From there, you run a query, you obtain a feature which points to an online resource, and then you can fetch actual data that you can work with. So how are these connections made? We have semantic links. So one typical example is you have statistics of receptor densities in certain brain regions. Then the link is really by the name of that brain region. You know? The brain region is encoded as metadata in the data set, and we can use that to actually find it and make the connection and attach it to a brain. Some data is linked to a coordinate, for example, in M&I space. That's the case for the microarray data in the Allen brain that was also discussed this morning. There we have M&I coordinates, and we can use these M&I coordinates to actually pull it into the system. And sometimes it's images. So we have an image, And then we can use image registration to actually, one of those reference spaces to actually link it in. So these are three pretty different ways of linking data into this Atlas ecosystem. And when you do this, a typical application would be that you have two different regions of interest, for example, two brain regions that you're interested in, and you do such a multimodal profiling of those regions for those regions of interest. And then you can, of course, run a statistical comparison of the features that you get, for example, densities of cells, connectivity information, gene expression values, things like that. That's what we are after. Then let's have a look now, finally, after a lot of talking, probably, let's have a look at the online viewer and see how that works. So I'm switching to the viewer here.
If you have a laptop, please feel free to follow if you're interested in. So because it's completely free, you can directly do it.
So if you launch the system, you should see something like this, and maybe Sebastian takes it.
Maybe you will be asked for a guided tour the first time you load the viewer. You can skip it, then it will ask you again later on, because this guided tour is.
Maybe show the link again.
There's a link to the viewer. Was it Go Viewer?
It's atlases.ebrints.eu/viewer. Here you go. It's here on the left. And you can add /go/human, but that's not strictly necessary. atlases.ebrints.eu/viewer. There we go.
Okay, so yeah, this is one of the typical starting views you will have when you start the Atlas viewer. So the complete concept is also visible here in this breadcrumb navigation. So here we have those different atlases which are included. So you can switch between the human atlas, monkey, Maxon space, Allen mouse atlas. And so here we are in the human space. Here you can select the different templates, so reference spaces. So you can interactively switch from here on to the BigBrain space, and Zebra will try to keep the position as good as possible. So there's a really direct transfer from the MNI to MNI 152 to the BigBrain reference space. And then depending on the atlas and also on the template you have chosen, there are the different parcellation schemes which are available. So here we have cortical layers, there are some areas, and there are also high resolution images, but this we will see later on.
We should mention that the transition between the reference spaces, which preserves the view point, actually uses a spatial registration scheme, which is driven by cycle constraints, and which was already discussed earlier today by our friends from France. So there's really a link here also between the tutorials. So
here we have those 4 frames, those different cutting directions. So the frontal one, sagittal, and horizontal one. Here's a 3D representation. At the moment, what we can see here are the probabilistic maximum probability maps. So we have sharp borders between the individual areas, but we can also, if we select one, so this can be done by clicking on the individual area. Then we'll see the probabilistic maps we have just talked about here from the primary visual cortex. And but this can also be done using text search. So if you already know what your area is called like, so in this case it was HOC1, you can also do it with a text search, or there's also a region tree. So there are different ways to select areas you would like to work with. So in general, the viewer here, the Zebra Explorer, is it's really helpful and easy starting point into the Zebra world, because everything you can see here is also available using ZebraPython. So at the end, it's working with ZebraPython under the hood, but here it's an easier visualization. So you can get an overview about the areas which are available, about the templates, about the atlases. But if you really want to create workflows and work programmatically with the data, then you should switch to ZebraPython, because there you can really code everything and create workflows and even include them in your study or in a publication. So in general, is it here we have those 4 frames showing the individual maps or an overview of all maps of the selected data. And if you select something on the left side, you have this menu here giving you an overview about details and descriptions of the areas of about the area you have selected. If you click centroid, it will bring you to the centroid of the area. So because sometimes if you have selected by the name, you do not directly see which map you have selected, therefore it's helpful. And what's also very important here, as I mentioned at the beginning, those are not only labels, so there are a lot of details available for all data sets you can see. So there's really a clean and clear meta information available. So every time you click on a map or here on the complete map set, there's a link to the knowledge graph. Can I move it? Okay. So this is now the knowledge graph data card for the complete parcellation scheme of the Jülich brain atlas. But such a data card is available for each individual map. So if you click onto something like this and go to the knowledge graph link, you will get an overview about the data set. There's a complete data descriptor giving you all details which are necessary to really work with the data set. So the scientific concept, the studies which were published to create the data set. There are also tools mentioned which you might use to work with the data sets, and it is really a comprehensive description of the complete data set. And you can also you can download everything. So the whole atlas is packed as one release package here for the 3.1 version. With a lot of MISA information about the terminology, about an overview, which areas were used, and about all maps which are included. So if you would like to use your own tools or whatever, feel free to download it, and everything is here. Also how to cite it, and yeah, a lot of details about it.
So are you able to download the actual region?
Yes. So if you select the region you are looking for, you will have the same button here, and will be directed to the knowledge graph and can download it as one individual map, or you can download the complete set and only extract the one you would like to. So
you can even do it here in the browser, but typical way would be that you go to the original dataset page, you find nifty files there, right? But also the browser here is you see here a download button. It also gives you the download, but it's a rather new feature. So in what always works is you go to the dataset page and download it.
And it comes in nifty.
It depends a bit on the data set. As I said, we attach here different resources, but usually they are nifty, right? We will show you also large files soon, like the BigBrain, for example, is of course not a nifty. It's a real huge data resource, but there are other ways to download it, and we will get to that. So the data is very heterogeneous. It's not everything a nifty that you touch here, but these probability maps are.
Okay, and another thing Timo already mentioned are those regional features. So again, we are here with the area HOC1 or V1, the primary visual cortex. And depending on the area, there are different features linked to this area. This linkage can be on a semantic, on a spatial, or on an image-based way, as Timo just described. But all features which are linked to this structure, to this parcellation map here, can be found here. So they are grouped a bit to make it not so crowded. For example, there are some cellular data sets. So we have layer-wise cell densities where we have extracted patches of images at 1 micrometer, which were manually annotated for the specific lamina. There are cell counts available, cell sizes. So it's really 1 micrometer data which can be downloaded here. But there are also profiles from the BigBrain which were extracted. They can also be downloaded or used in siibra-python. There are further marker structures. So they're really grouped depending on their modality and on their structure. And the receptor densities are here. Some of those features are more deep embedded in the viewer. So sometimes you can really see the profiles or even patches of receptor densities. Sometimes there's only a description and a link to the knowledge graph so that you can get the raw data. But everything which is listed here can be downloaded and used. And the amount of features really depends on the area. So it's not the same in every area here. Okay, one.
By the way, regarding the cell densities that you see here on the left, we will get to this in a bit more detail in a couple of minutes. And also, we also have a slide describing these data sets in a bit more detail.
One functionality, which is also quite new, but I really like it. If you have a coordinate from your own study, giving you an activation in somewhere in the brain, and you are not really sure what is at this location, you can directly insert those.
Question.
Yeah, let's.
Okay.
So you can simply type in the coordinate and select the point. So now we have the centroid of V1, so there should be V1, but you can do it for every position. So if you have your own coordinates, your own publication with some specific features, you can directly see what is there and what kind of map should be there at this map, at this position. And here you can also see the concept of probabilistic maps. So here we have the centroid of area HOC1, so it's extremely high probability of being area V1. But there's also somewhere in the zero behind the comma or dot a possibility of being HO2, so secondary visual cortex. And this is at the end the inter-individual variability. So if you go to a point where more areas in surrounding, it's quite often the case that you will get several results. But at the end, we think this is the best way to describe a position, because there is always a bit of possibility of being in the neighboring area, and this should be kept in mind. It's not always one-to-one assignment possible for every position. And what's also one of the things Timo mentioned was the switch between the different reference spaces, which is also included here in the viewer and also in siibra-python. So again, now I've selected area 4P, which is the primary motor cortex, and I've moved to the centroid coordinate. And if we now switch to the
Let's say
okay, so yeah, here we are now at the centroid of area 4P, but we are in the resolution of the 152 reference space, which is about 1 millimeter. And of course, this resolution is not sufficient if you really want to see cells. And therefore, if we there comes in the BigBrain, and if we now switch to the BigBrain, and so siibra tries to keep the position as good as possible. There's always there's always a bit of variation, but mainly you are here now in the primary motor cortex, and of course you still can't see that. So this is the BigBrain, which consists of 7,000 sections with 20 micrometer in plane resolution, and here maybe to give you an overview, it's like in the viewer before. We have different cutting directions. So here is the original frontal cut direction of the BigBrain, and those are the reconstructed cutting direction of the BigBrain. So those are completely virtual. This is the only real existing cutting direction, and those were reconstructed. The layers here were published by a study from Konrad Baxbühl. There wasn't he automatically find the profile, the lamina based on gray value profiles, and this is working most of the time really quite well. And what we also have here below this 20 micrometer resolution is that we do have higher resolution data, which is also available. It's available here in the viewer and also with siibra-python. So again, if we select those regional features, here we can see different high resolution data sets which were anchored into the BigBrain reference model.
Maybe I can say something here. Here you see exactly the distinction. Previously, we saw features that were linked to the brain region as a concept. So we clicked the brain region, we saw the probability map, and we saw, for example, receptor density, cellular features that were linked to that brain region. What we see here is a spatial search. So we find here data that is defined by coordinate or by a bounding box in a reference space. So this is exactly the second path now where through coordinates and by being in a certain coordinate space, we find data.
And now here, let's do it a bit bigger. So now we really have 1 micrometer data. So this is the ground truth for the data sets we have used when we created the individual maps of the different brain areas. So 1 micrometer really means that you can see each individual cells. And at the moment, there are 150 sections available here in the Zebra Explorer. So it's roughly every 50 section which is anchored and can be used. So you can extract data sets here from the viewer, but you can also interact with those 1 micrometer data using the Python library if you really want to create your own workflows with the data sets. Okay, is there anything more you want to add for the BigBrain here at this point?
Möchte ich noch eine Region zeigen?
Okay.
Oder Region zeigen, dass wir sehen kann?
Yeah, it's we want this.
Yes.
Okay. Sorry, it's a bit slow here. Okay, what we are currently looking for is the border between V1 and V2, because it's the very beginning. I showed you this one slide where the very obvious border between area V1 and V2 is. And this one can also easily be seen here in the BigBrain. So here we have the alkaline sulcus at the occipital pole of the brain, and okay. Ja, ich glaube, wir müssen weiter rein. Okay, so here we are. So this is exactly, it's not exactly the border from the same slide, but it's the same position. So here you have the calcarine sulcus with area V1 showing this very high cell density in the lamina 4, deep in the cortex. And here we have the secondary visual cortex showing a completely different cellular architecture, and the border would be located somewhere around here. Unfortunately, it's a local artifact, but I hope it's convincing that there are microstructural differences between adjacent areas, and this is a very easy one. Of course, there are more difficult ones, but at the end, those differences build the basis for our maps. Maybe something we can also show here before we show the programmatic approach for the okay, sorry. Something which is also included here, and this also connects to the thing Michael Holovitz this morning showed, and there are different plugins already included in the online view and also in the Zebra Python library. For example, the Zebra UJEX tool. this is exactly the one Michael Hollowitz showed on this one slide, where the Jülich brain maps are used to extract data sets from the Allen Brain microarray data set. So because this is also built on the idea that we need reference spaces. So they brought their data set into the MNI reference space with specific coordinates. We have brought our maps into the same reference space, and by having them in the same space, we can simply use the maps to extract those tissue samples which are helpful or which are of interest. And yeah, once you have selected them, then it's only statistics to find differences between your areas. And this can be done at the end with every individual volume. So if you have volumes from functional studies or whatever kind of metric study, if they are in a reference space, you can bring them into the siibra-python environment and having them there directly extract data sets from the Brain Institute and really work with their data as well. So it's a different way to interact with the data. But this is exactly the idea and the way we would like to develop this atlas, so that we really bring in different modalities, different institutes, and because it's so complex, it can't be solved by one institute or by one modality. So we really have to work together and to bring the stuff into the same world and learn to interact with each other. And yeah, that's why we think such a multimodal approach is the way to go. And yeah, we start implementing it here. Should we switch to
maybe go back to slides. So we have seen this principle now. We have also seen that the things we are touching here in the viewer, behind everything, there's usually a curated data set. You have seen that. And we have also mentioned that you can switch between reference spaces such as BigBrain, MNI space, and the surface space. And you can do this based on the level of brain region maps that are corresponding to each other in the different spaces, but also spatial transformations. We have mentioned that the transformations that we have linked in here in the background are based on cycle constraints. See a publication for this. And we have seen how we can zoom in to find individual histological sections and work with that. Maybe there's also a good time if you have questions. So if something was unclear or if you particularly have questions about this, would be a good time to cover this now. Yeah.
So is it possible to map structural phenomenon within events, for example, another like HCP datasets within IRA with the siibra-python? So it's possible to map this kind of that structural phenomenon to brain regions?
So the way we have done this currently in Zebra is, I think it should be even here. So
there already is some data set.
Yeah, there is some of that data there, but it is in a way pre-computed. Actually, it is everything here at the moment. That's interesting. Well, so what you currently find are connectivity profiles for brain regions of the reference populations we have, which have been computed based on such data. So which are based on statistics of streamline counts, streamline lengths, also functional connectivity in the system. But they are pre-computed. So they are also data sets in eBrains, and someone took the postulation, ran the computation, and computed these profiles per brain region. Basically we have linked region-wise connectivity maps, connectivity matrices. Something that we are somewhat working on is to make that more flexible so that the system actually knows the computational processes of grouping the individual streamlines per brain region. So that actually, if you use a different postulation here, the system would be able to still deal with that. That's not currently the case. Yeah? So currently we have some pre-computed data sets in there, which are indeed based on HCP and other cohorts. but it's kind of static, that it has been pre-computed on the parcellations that we have here. So if you release a new version of the siibra-python atlas, for example, which we do every now and then, we actually need to recompute these pipelines, publish a new data set in order to have the connectivity available again. And I think that is actually also the reason why we didn't see it here. Now that I talk about it. So if we go to the earlier version of Jülich Brain, and then we select brain region. Hope I'm not. So you see here connectivity, yeah, and we see here streamline counts, yeah, and what we see here is now this data sets based on the HCP cohort, or which where we took the streamline counts and computed profiles with respect to the current parcellation. And what you see here now is a coloring of the brain regions. I can use a log scale, which makes it a bit more explicit. Coloring of the connection strength with respect to the selected region, right? So we started out here with the primary visual cortex. So we see here a row of the connectivity matrix, if you wish, which is just center there. A row of the connectivity matrix, which refers to the primary visual cortex. Yeah, we could even travel here. So you see here that, of course, the secondary visual cortex has strong connections. That's what we see here. I can even click here and then to go there. And of course, I can I can select it and move on this way and explore the connectivity. And these are also data sets. Yeah, as I said, even here you find a link. And you see here that this is based on a published data set where the connectivity has been computed. Oh my God. As I said, there's this week, there were several migrations of virtual machines happening in the background. So yeah, here we go. Yeah, you see that's also a data set with the UI, with a data set descriptor, all that's these things which describes this, which has been integrated to the system. So if you go here to get data, what you actually see here is there's a bunch of zip files for different parcellations. You see the Jülich brain parcellation here. And in that zip file, you find a connectivity matrix in TSV form and so on. The system does that for you, right? So the system knows how to open that zip file, get that data, and present it to you already. And you could also here right away download the CSV file in the viewer. So it basically gives you a shortcut and allows you to explore that, but it still has the link to the data set. Yeah. Hope this answers your question.
Yeah.
Talked a lot now because we left the connectivity out in the presentation, but I'm happy to talk about it as you see.
No, I'm actually interested because I actually use that data set to export some structural connectors for hippocampus that I use as global connectivity for specific brain activity simulation. So that's why I'm interested. And also, is there a way to also include like region mapping of a region using probability maps and also mixing these structural connectors?
You can do that, yes, but here in the viewer, the viewer does not support such a thing at the moment. You can actually, you could use the Python library to do such things. Yeah, yeah. At the periphery, we can touch this now as we look into Python. But this is definitely something where we would need to do some coding. Yeah, but I think that's possible. As I said, we are quite interested to make this more dynamic with the connectivity. In the past, we didn't have a lot of time and funding to go very deep in that area. It's very interesting. We can also discuss more if you like. It's interesting. Okay, there's more in the chat. It's always you are monitoring the chat, Susanne. That's good to know. Okay, good. Yeah. Let's just have a little look in the Python client. As I said, it's not so much time today to go into deep detail there, but I have prepared a few tutorials. So here's a tutorial from our last winter school where we'll show you a few things, and then I would also like to show you how you can access microscopy data with the Python client.
Ja, und vielleicht nochmal den Link mit dem Tutorials drauf sind, ja.
Ja, den Link könntest du mit im Chat.
Okay, so ich mache alles, was ihr sagt. There is first of all for the Python client a good entry point is the read the docs documentation here. I just put it in the chat as you see, and there you find some documentation, API documentation. Especially, you also find a step-by-step guide where for many things that we are looking at today, you find. Let me just. Well, let's go with something with the maps. For example, here for accessing a brain parcellation map, you really find here a short documented code snippet, which shows you how to do certain things. We will go into this with in a minute. Just to tell you, in the documentation, you find such code examples. They are tested automatically when a new software release is done, and you can download here a Python notebook or the Python code, and it should be working right away. And it's usually small ones, and they show particular aspects what you can do. That's a quite good way to dive in. And then we have this GitHub repository, which today, so Zebra tutorials. If you Google for Zebra tutorials, it will take you there, where we also prepared a few longer notebooks. And you also see here's one from the last Cyber Winter School, which we'll be looking at now as well. And you also find links to run them on. I do not suggest that you do it now because I have seen some of them not working due to the migrations that happened in the past days. But we will fix them. So after this workshop, everything should be running there. Okay. So the important message is really that the Python client does everything we've just seen interactively in the viewer. So you can really literally do everything we did there. You can do it in the Python client and more. It's of course a bit tricky to translate these very interactive things into something programmatic. And I try to explain to you some of the ideas. So the package is called Zebra. Of course, you would export this package. And as we discussed, there are some key concepts that we are always dealing with. Parcellation schemes, reference spaces, parcellation maps, and brain regions. These are important concepts, also data features. Zebra for these has modules which have their own registry, if you wish, of pre-configured content. So an important thing to understand is that the software itself has no content. So if you just take the software, there would be no maps or anything because the software has no data. But the software actually pulls a default configuration from GitHub, which has definitions of all the content that can be connected. And per default, it loads such a configuration with all the data I'm showing here. But you can also create such a configuration yourself or modify it if you take the Python client. Of course, the online viewer that we just saw uses a default configuration and you cannot modify it. But if you work with a Python client, you can actually point it to a custom configuration and include other content or modify it. But what it does in the background, when you import the package, it really pulls a default configuration from a GitHub repository that we are maintaining, where all these things are described, and you can work with them. Then it populates the different modules. So there is a registry of predefined parcellations, which you can then browse maps for these parcellations and regions. So usually what you would do is, for example, write zebra.parcellation get and write a few keywords, for example, Jülich 3.1, which would give you the parcellation scheme of Jülich 3.1. As we discussed, that's not yet a parcellation map. That's a parcellation scheme. That's a semantic object. At that stage, there was no data loading or anything. That here's a very simple step. It more or less just loads the region tree and the names of those regions. If you want to access a parcellation map, that parcellation, you need to provide a reference space because we have maps in different reference spaces and these are different. So in order to obtain a map, we basically request the map from the parcellation scheme. We provide a space. Explicitly, we would write space and something long, but we can also use a short keyword, MNI 152. And we provide the type of the map. If you don't provide it, you just get a labeled volume, which is probably the most common thing. But we have shown you here these probabilistic maps, and you can also request those by providing as a map type statistical. So in this case, you would get the probabilistic maps, which is one map per brain region, hundreds of them in that case. Yeah, if we would leave this keyword out, we would just get a single labeled volume. I just put up one slide again, which is at this stage, this one here. So we're actually going exactly through this hierarchy here. Yeah? We choose a parcellation with regions, then we have it attached to its space to arrive at a map. Yeah, from the map, if we actually want to get some image data, there is a fetching step involved, because everything we have here is a pointer to an online resource which describes everything. In order to actually get the data, there's always a step that we call fetch. Yeah, so Zebra will only actually load data if you request or use it. Until then, it just knows where to get it, and that's exactly happening if we look again in the code. So we got access to the map. This still did not load the mapped volume, but at the stage where we say fetch, now we actually get an image. It will actually get an image from the online resource, and since these are probability maps, we constrain the fetching to a particular region, because every region is a single volume. Yeah, we don't want to load all of them. So now for the left area for A, which is from the motor codex, I hope. That's happening here. Let's look a bit further. You have seen already we have this module of predefined parcellations. If we print that, we see a list, what's in there. So you see here's many different versions of the Jülich brain atlas. There's an atlas of functional modes. There are fiber bundle atlases from Neurospin here, and also, of course, parcellations of other species. We can access those. We can also access them with tab completion instead of keywords. We can, with all the elements that we actually get here, we also can access metadata. Yeah, since in the background it knows the online resources, we typically find also properties like descriptions, publications, DOIs here that we can access right away in the Python client. Once we get to the level of regions, regions are still semantic objects. And in fact, in Zebra, a region is a little different from a parcellation scheme, since parcellation scheme in Zebra is just the top node of a region tree. Yeah. So whenever you have a brain region, it is a tree on its own, because it can be the parent region of additional child regions. And we can visualize this here as a tree to have a look. So if you look at Jülich brain, you see that's really a pretty long region tree with many areas mapped. The recent version has almost 300.
227.
227. And there are two granularities available, so there's even one with a little bit more in the two hemispheres. And you can search those regions here also by providing keywords. In Zebra, very often you search for things, and we always distinguish two functions. One starts with get. Get assumes you know the keywords very well, what you want to get, and you know there should be a single response. So when you say get region, Zebra assumes you want to obtain exactly one. So it will complain if the keywords are not sufficient, or resolve it to the parent region, and if not, it will raise an exception. There's also find. So if you say find regions, you get a list of everything that matches. That's a repeated pattern that you find all over the place. So if we get the region amygdala, it will not return a list. It returns, in that case, the parent region of the amygdala, which has children as well. So it's a region tree as well. We have already seen how we can fetch parcellation maps. At the stage where we apply fetch, Zebra always tries to take you out of what we call Zebra land and give you an interoperable data format. So if we fetch from a resource that reflects an image, you will get a nifty image object. If you fetch from a resource that has tabular data, such as cell densities or things like that, you will get a pandas data frame. So usually when you say fetch, you get out of the Zebra software and you have something that is very interoperable with many software libraries. In that case, so what we get is a nifty image. You see that here. And the nifty image, you could use that right away, for example, with NiLearn, which is a very popular framework, and just plot it. So you don't need Zebra for that. There's software that can do it. Same, if we fetch a particular region from a labeled map, we get a mask as a nifty image, and we can plot the mask. if we fetch a region image from a probabilistic map, we get a nifty image, which is such a statistical map, also easily plottable. And we can also request a map in a different space. Currently, we only use the M&I space. If we supply here a different space, the free surface space in that case, which is a surface-based space, we also get a map, but that's not a nifty image. That's actually a dictionary with a surface mesh. So fetch here returns us a surface mesh, and this surface mesh, we can also use it in that case again, NiLearn to display and use it. So the visualization here is not based on Zebra, it just uses existing software libraries. Are we supposed to end at the quarter past? Sun? Yes. Okay. These were maps. A very, very similar pattern is available if you deal with spaces. Yeah. So you have a question?
Yeah, so for example, using this fetch method, is it possible to extract from siibra-python a surface mesh of big red region and also extract the probability maps, I don't know, as a matrix or any kind of data structure?
So you cannot mix it arbitrarily. So typically, if you have a particular entity, there is in most cases one type of data behind it. That's not always the case. Sometimes there are multiple, but usually it's one type of data behind it, and then you just get that. You can use fetch in BigBrain. I will show that. You can also use fetch to retrieve surface meshes in BigBrain. They are pretty huge. In fact, the newer version of Zebra will be able to cut them to volumes of interest. I think the current one can. We would see that actually in a minute. But that's a bit experimental, partial meshes. You can also retrieve brain region maps in BigBrain. There's actually also an example for that in the documentation. We don't have the probability maps in BigBrain yet. So that these are always in a nice space at that stage. Yeah. Which is not a big, doesn't make a big difference, but if you're interested in that, I'm happy to chat with you. Yeah, so for reference spaces, it works the same way. Yeah, for brain maps, we had the parcellation scheme, which was a semantic representation. Then by supplying a space, we have a parcellation map, which is a spatial entity, and by using fetch, we actually get some data. For reference spaces, it's similar. The reference space is first of all, it's a semantic concept. It has a name, it defines a coordinate system, and so on. Then we can, from there, get a template, which refers to a particular anatomical image representing that space. And we can use fetch again to actually pull that image from its online resource. That's happening here. So the BigBrain, which we talk a lot about at this workspace, at this workshop, is a reference space in Zebra. So you can actually access the BigBrain as one of the spaces. That's just a semantic object. You can get a template that's analogous to get map for the parcellation maps, which defines the actual template. That's the BigBrain volume in that case. And then you can fetch from BigBrain to retrieve a nifty file. Now BigBrain has a size of 1 terabyte. The actual data set is an image of 1 terabyte. So if you use fetch here, you would actually download 1 terabyte, and you don't want to do that, right? That's usually you will not have so much space on your laptop. So what Zebra is doing when you run fetch without arguments, and that's an image, it actually does a check how much data you would get. And in case of the image, it will realize that's a terabyte, and it will actually downscale automatically. It will fetch a lower resolution, which leads to feasible download size, and it will inform you about that. It will tell you, okay, it's a huge one. I will download only a resolution of 100 micrometer or less, which leads to feasible download size, and here you have it. So what we get here is a nifty image of the BigBrain, which is feasible for download and can be displayed. Of course, often, yeah.
Can I specify like a region to only download?
Yeah, that's exactly the next step. Yes.
And get the probability maps in a vertex level?
Yes, you can do that. It's another probability, but that's exactly the thing. So if you are interested at a higher resolution, the Python client here requires you to do the very same thing you would do in a viewer. In a viewer, you don't see the full resolution when you're zoomed out. Yeah, that's not happening. To see the full resolution in the online viewer, you zoom in, right? And that's the same thing here. So usually you define a bounding box, or you define a brain region which has a bounding box. This gives you a smaller piece, and you can then fetch this smaller piece at higher resolutions. And I think this is also shown here. So the way we did it here is, first of all, we define a point in space. In this case, it's the M&I space. We translate the point to BigBrain space, which is the case here. So that's something in the motor cortex. And then I'm doing a bit quicker because of the time. Of course, we could talk much longer about that. Let's just go a bit more to the bottom. Is it actually happening here? It's happening here. Yeah, here we go. So you define a bounding box, yeah, between 2 points in the BigBrain space. And what you can then do is you can also run fetch as previously, but you can provide as arguments this volume of interest, which is a bounding box definition. And you can then specifically request a full resolution, 20 micrometer here in that case. Yeah. And then what you get is again a nifty image, but it's only a partial volume now at a much higher resolution. This nifty image comes with appropriate spatial metadata. So it has an affine matrix attached, which actually knows where it belongs in the whole brain and so on. So you get here a full resolution chunk of the big brain as a nifty file with spatial metadata that places it appropriately in the whole brain. And you can actually co-visualize directly these two things, the lower resolution of the whole big brain and the higher resolution of this particular chunk of the big brain. I'm not sure if I'm doing that here, but actually in the here in the online documentation where you have these examples, there's definitely access BigBrain high resolution data. Here you see already. There is here a code example. It's exactly that which shows you that. Yeah, so we get the lower resolution whole brain. We get a region of interest at full resolution, and you can simply co-visualize them. Here you see that it's so tiny, but the little red thing is the 20 micron resolution chunk, and it's properly placed in the whole brain. So you can do this, and you can, of course, you can download the whole big brain. Zebra allows you to do it, but that takes a lot of bandwidth. All right. As a last thing, what I would really like to show you in the last 15 minutes is how to sample high resolution data. Sorry for the forth and back here. So we already showed you in the viewer cell densities. And what we actually did to get cell densities attached to the atlas is we extracted cortical image patches like this one from the original 1 micron sections of BigBrain. Yeah. So we know where they belong in BigBrain. We have, you see here the dot. We know where they were taken, and we have chosen hundreds of them, which have a nicely orthogonal cutting direction, good tissue quality, and so on and so forth. They really look like this. Here's one in the motor cortex. You see how this looks different. Here we have the big cell.
Beats giant cells.
Big beats giant cells that you would expect in the motor cortex, for example. You see that here. We then used a cell segmentation AI algorithm to extract at high precision cell bodies in these patches. Actually, tomorrow, Eric Upschulde, he's here at the workshop. He will also talk about cell segmentation. So Eric is around and you can chat with him. There's a lot to say here. I'm not going into that. And this allowed us, and we also delineated the cortical layers precisely in these patches. And this allowed us to extract statistics of numbers of cells per area, so cell densities in the different cortical layers in the different brain regions. Here, in that case, what you see here, based on 120 such patches. Since the cell segmentation gives us the contour of the cell bodies, we can also group them by size. So what you see here are characteristic histograms of numbers of cells per region found in the different cortical layers according to size. And this gives you quite characteristic fingerprints of the different brain regions. This is available through Zebra. I'm just showing this here from a more conceptual point of view. So these here are the positions where we have measured such patches. And there is a pretty good interest in the community in this data. So we are currently working hard to have more of them. So the data set will soon be extended to almost 1,000 of these patches. That's in the works. So you see the distribution here. So the ones at the top already are accessible to Zebra and published on eBrains. And you saw that already in the viewer a couple of minutes ago. Now, what we did here to extract these patches, of course, we would like to give these tools in the hands of the community. And actually, this is possible to see. And this is the last thing I would like to show you, because it's also something that I love to do. So here's a piece of code based on Zebra, and this tutorial is also on the Zebra tutorial page, which actually takes you all the way from just spelling out the name of the brain region down to getting such an image patch automatically in a reproducible fashion at 1 micron resolution. So let's have a look how that works. I'm just importing Zebra again here and a few other things, and I increase the size before someone reminds me. Okay, so I just specify the name of region here, which is just only the name, right? So I'm here interested in the motor cortex, so I take the name of a region 4P in the right hemisphere. I just define those words. That's nothing else at that stage. And then I do what we have already seen. I request the Jülich brain postulation, and from the Jülich brain postulation, I find that region just by putting these two words together. So as a region, I should, in that case, get a region in the motor cortex in the right hemisphere. Then I request the maximum probability map from the Jülich brain in MNI space. So we are at that stage not in BigBrain space, in MNI space, and I fetch the. That's actually not correct here. It's not probabilistic, but it doesn't matter. I actually fetch here the map of that region in MNI space. Yeah. So at that stage, I know where the region is in MNI space. Yeah, because I just fetched the map, and I have here now a nifty file with that map. I can plot it here. So you see that's here where you would expect it for the motor cortex. It's just a nifty file. Now what we want to do is we want to find histological data that is located at the same position, and we want to extract a cortical patch which is nicely oriented in the cortex, shows all the layers, and has a full resolution, yeah, and is with high probability in that area. So what we want to do is we want to find a brain section, big brain, which is roughly here where we have the high probabilities. Then we want to intersect it, so we get that part, and we also want to find out where the cortex actually is, so that we can get the patch in orientation of the cortex and not somewhere where we have only partial cortex or background or something like this. And this notebook is doing that, and it's actually not difficult. What we do now is we actually put the nifty back into a volume. Why? Because in Zebra, the volume has also the name of the reference space attached and so on. This is important because we work with different spaces. So we basically just containerize it again into a volume. Then we can use this volume to query features in Zebra. Features query is always the same thing, zebra.features.get, and it gets two parameters. The first parameter is a representation of some form of location in the brain. In that case, it's a volume. Could also be the name of the brain region. And the modality. And since we are looking for brain sections, the modality is here cell body stain section. What Zebra is then doing, actually, it searches all the cell body stain sections that intersect with that volume. And that's interesting now, because the volume that we provide here is in the MNI space. Yeah, we have obtained it by specifying the name of the brain region in MNI space in the Jülich brain. The cell body stain sections that we are interested in are anchored to big brain. That's a different space. Yeah. So what's happening in that single line is that the system actually translates the bounding box in MNI space of that region into the big brain space by calling a software that uses Salker constraints and nonlinear deformations. So it automatically warps the bounding box into the big brain space, since it doesn't find any of those data features in the MNI space, but it finds them in big brain space. So it automatically warps the bounding box there, intersects the bounding boxes of the images with the brain region, and gives me a list of those sections. Yeah. What I do here is I just take, because there will be a couple of them, I just take the one in the middle. It doesn't matter. We could also take the first one. So what I have here now is one such brain section which intersects. Now this is still a whole brain section, right? It still doesn't tell me where the patch in the motor cortex is. So what we do now, I also request the cortical layer maps as a parcellation from Zebra. You have seen the cortical layer maps in BigBrain space previously. So I get this map in the BigBrain space, and I create a 3D plane from the brain section that I'm interested in, and intersect this 3D plane with the cortical layer maps. This gives me the contours of the cortical layers in that particular brain section. Yeah. So there should actually be here's a visualization of that. So actually what I get then is, for that brain section, the contours of the surface meshes of the cortical layers intersected with that image. Yeah. So now I know where the cortex is, right? I could, for example, take the contour of the layer 4 boundary, which is pretty much in the middle of the cortex, and then I'm already in the cortex. I could also use the other contours to define the direction, so I know actually how is the cortex oriented. Yeah, so this gives me orientation. Now I still have to find a place where I'm very likely in the motor cortex of the right hemisphere. So what I then do is I evaluate all the points on the layer 4 surfaces with the probability map of the motor cortex. Yeah, Zebra can do that for me. Here again, it translates automatically between the spaces, so I don't have to care about this. The probability map is an MNI space. The points on the layer 4 surface are in BigBrain space, and Zebra resolves that for me. So what I get are the probabilities then, and I just take the point with the highest probability. Now I have a point on the layer 4 surface in that histological brain section, which is with high probability in area 4P of the motor cortex. And I also know the cortical layer boundaries, so I know the orientation and the thickness and so on implicitly. And this is the last thing I'm doing here. I'm actually building a bounding box here, and I also extract the surface normal from the corresponding vertices on the layer 4 surface. That's a few lines of code. It's a bit hard now to explain in a few minutes. But actually, I get these points here, which are all close to the highest probable point of area 4 on these different layer surfaces. So I actually now get a nice line where I want it to be in the cortex from the inside to the outside. And I can use this line to build a patch. And this patch now, that's the last thing what I'm doing, this patch I can use to extract at the full resolution of 1 micrometer, a patch from the brain section. And only here it starts downloading image data. Everything else was just working with bounding boxes, you know, and spaces. There was not really a download of image data involved until now. But at that stage, I'm really using again fetching mechanism, and I'm downloading at full resolution a section. And what we see here is a rotated patch, which is again a nifty file, although it's 2D, and it has spatial metadata, which allows us to position it exactly in the BigBrain. Yeah? So by running this code, I have a reproducible way, which takes me from the name of a brain region down to a 1 micron resolution cortical image patch with cell bodies, which is appropriately placed in the BigBrain and also has in the metadata a position in MNI space. Yeah. So here's the final image. Yeah. And of course, there were some stages where you could introduce sampling. For example, instead of just taking the most probable point on the layer 4, I could take a few of them. And this way sample multiple such patches for doing a study. Yeah. This was probably a bit of a stretch now at the end, but what I'd like to show you is that there are really ways with a couple of lines of code to do such a complicated chain. And it is really a complicated thing. If you would like to do this without such a software, you would really have to get your hands into a lot of different software packages and online resources to actually get to this data, including to download very huge nifty files and then cropping them yourself. Zebra is doing all of that for you, right? So you didn't have to do any cropping. There was no need to download a large data set. In the end, we just got this simple patch, but still we were doing all the computations with the large data.
Would you say that you need this sort of code a lot more for this very precise localization, but if you go for just a generic region, like some part of the visual cortex are just being downloaded from the software?
Yeah.
It's easier.
Then it's much easier, right? Yeah, yeah. So what you can always do and what most people would do is you click the brain region, in the viewer the sidebar opens and it tells you there is already a data set with cell densities for that region, which we have in that case prepared. And it has a DUI and you download it and you have already then an Excel sheet with these numbers. Yeah. But we actually use this mechanism so it's reproducible and you could sample other data sets here. For example, if you have your own brain structures and a different methylation, it will not be that easy because you have probably a different name for that brain structure and you would still like to sample microscopy data there. Yeah. And then you could still use such an approach. This notebook has a lot of code around it. There's a lot here about visualization and descriptions. The actual code to only do the fetching, I am preparing a manuscript at the moment and have it in a figure. I think the actual code is 15 lines to actually get the patch. So there's much more here because I visualized steps. All right, I think with that we are at the end. Yeah, quarter past. Of course, we are still here. If you have any questions, or you could also check the chat. It's been fine there. Yeah, there's another question there.
Yeah, so there are other methods within Siibra that allow you to interact with the knowledge graph and extract functional and connectivity data in a format that is ready to go to use in the virtual brain. So are these methods still under maintenance or they are not outdated?
I would need to have a look with you. They should work. But as I said, right at the moment, there have been a few maintenance things and a few updates. It might, if you have trouble, I'm happy to have a look. But they're definitely maintained. Yeah. With the connectivity in particular, what you might have encountered is that there is a new version of the Jülich brain oscillation. And as I said, for the connectivity, we need to recompute from the original streamline data. And this recomputation still hasn't been finished. So there's usually a certain delay until with the new oscillation version, until all the linked data that has to be recomputed is recomputed and published. And that is the par excellence example is the connectivity, because most of the other things don't need such maintenance. But the streamline statistics need it. So in that case, if you currently use Siibra and you don't pull the older version of Jülich brain, but the most recent one, it will not find connectivity profiles because they are still being computed and published. I hope that to be finished in a week or so. What I learned from this is we should actually there should actually be a message, because it's probably a bit hard to understand that. Yeah. Since Siibra will always give you the most up-to-date version of the oscillations, but it will not tell you that this might prevent you from accessing connectivity or so if they are very new. Yeah. All right, and thank a lot for your attention.